excels in chromatography and mass spectrometry, focusing on food safety and pharmaceutical analysis. Her expertise includes multi-residue method development and identifying genotoxic impurities. She has a rich portfolio of over 25 applications and publication notes. I'd like to invite Dr. Soma Dasgupta to the stage. Over to you, ma'am. Pointers here? Yeah. Yeah, you can just use this. Okay, left and right. Thanks.
Ma'am, sorry, your phone.
Thank you so much.
Thank you. Well, hello everyone, welcome back after the lunch, and I hope you had a good break in between. So let's hope that whatever we built on in the morning, now we will be delving further into that, and I will be talking mostly about the emerging trends that we have in analytical testing. So before we begin with the session, I would like to extend my heartfelt gratitude to Ms. Sarika Agarwal and Mr. Ramesh Agarwal, the entire FoodWorks team, the dignitaries over here. It's an honor to be present amongst all of you with such esteemed colleagues and engaging audience. And thank you for this opportunity that we are able to talk to you about the emerging trends which are there in the analytical work. So without any further ado, let's start with the sections that we are going to talk about. So before we speak more about the solutions or the technologies that we represent, I would also like to give you a brief overview of who we are. So we at Agilent represent a company of more than 18,000 employees, and we are a premier institute which works with our focus in advancing science and delivering integrated solutions for instrumentation and workflow solutions. So when I say workflow solutions, we basically mean the testing requirements which goes with each and every regulation and the type of food matrices or the type of analytical world that we look at. So we are not limited only to food testing. We also work with the chemical industry, the pharmaceutical industry, the academic industries as well. In terms of products, we have more than 50 different kinds of technology platforms which spans across gas chromatography, liquid chromatography, mass spec, spectroscopy, cell analysis, and so on. And if I start counting all the platforms that we have, the different models that we have, probably that would end my 20 minutes over here. So we'll move on to our presence in India. So in India, we have a diverse team where we have sales and support people along with technical people who work for all the customers that we have across India. There's a centralized logistic hub. We have Center of Excellence in Mumbai, Bangalore, and in Manasa. We have a global business operations running in from India. We also invest a lot of our time into developing applications and workflows, supporting our customers both directly and indirectly, and also try and make a bridge between the global team as well as our Indian customers as well. We run into a lot of important collaborations with premier institutes like IIT Delhi, FSSAI, IISc Bangalore, ICT Mumbai, USP India, EIA Kochi, and NRC Grapes as well. We have regional offices, services, and sales center in India to help and support our customers with pre-sales requirements, post-sales requirements, and continued support. So in terms of our presence in food or the safety and authentication, Agilent supports the testing and surveillance of the supply chain to ensure that the world's food supply is safe and wholesome. So what it in other words means is that we help you comply with the testing requirements that you are needed to do as per the regulations, and also to maintain the quality of the produce that you have. So that involves solutions in terms of instrument, that involves solutions in terms of workflows, and that also involves solutions in terms of the softwares, which includes LIMS-based softwares, or it also includes, in the pharmaceutical sector particularly, the 21 CFR compliance requirements. We also collaborate with many of the thought leaders to develop newer workflows and work on the newer trends. In Agilent, we strive to develop complete solutions which will involve food tasting, testing, safety, quality, and authenticity. So our major focus is to develop a complete solution which is based on research, development, and quality assurance and compliance solutions. And all of this is also integrated along with Agilent cross-lab solutions, which include instrument service, the core servicing which is done, training and consultation with operational expertise, enterprise lab management, along with consumables like columns, the other supplies that you require to run your instrument seamlessly. So with all that introduction about who we are, what we do, let us also talk about our current focus and importance that we give to the current requirements into the food testing. So the four key areas which are now the talk of the town is ethylene oxide, which all of you must have seen across in the newspapers, various media channels, publications everywhere. PFAS, another emerging trend. Then polar pesticides, which has been there for quite a while. And also the last but not the least, which is the scarcity of helium gas and thereby switching over analysis to hydrogen-based carrier gas. So we will talk in detail about all of this in the upcoming sessions. So first we'll talk about ETO. So I will talk only from the perspective of the instrumentation and the workflow solutions that we provide. So ETO, as you all know, with a brief introduction, ETO is a fumigant and has also been identified as a carcinogenic, mutagenic, and toxic compounds. There are regulations which have come into force to maintain a safe produce and also control the amount of ETO in several producers. So as a response to that, when we had our customers, when we had our users coming back to us asking for solutions because they were facing a lot of challenges with establishing the workflows for ETO. Why? We will talk about it. But as a response, what we did was we developed workflow solutions, and there are 3 different workflow solutions catering to 3 different requirements. So some of the screenshots are shown on the slide itself. So these are 2 applications that we have. One is which is compliant with the European regulation, the methods which have been set up by the EURL. So the first application note on top, which you see, is from it is also from with one of our partner labs, along with our global team in EMEA. So they have worked and come up with this method. Then we have our in-house methods which we have developed in India, where there were customized requirements from some of our customers where they wanted to minimize sample preparation. So basically, what our customers wanted was, let us have the sample, we'll put it into the system, and I want the result. So they wanted it to be as easy as that, and we did listen to that. And with the help of our colleagues, we developed a method and a workflow solution with a headspace system along with GCMS to develop a workflow solution for ETO. Now, why do we need so many different kinds of solution? What is so difficult about ETO that we have so much of talk around it? Apart from the safety concerns, the major problem, if you look at it from the perspective of an analytical chemist, is that ETO is a very small molecule. So it has a very small molecular mass. It has an extremely low boiling point. The boiling point of ethylene oxide is 10 degrees. So literally, if you bring a glass full or a container full of ETO and keep it at room temperature, it's just going to vaporize. So when it is in sample, you can understand that even if it has been in the sample, then it is so difficult to extract the sample and then inject it onto the system. Plus, there is also a natural metabolism which is going on, which converts it into other conversion products. So which is why there is also a separate AOAC working group that works on harmonization of the methods and the analytical requirements to measure ETO. In this working group, Agilent has a representative from our European team, and we are also in constant touch with them, and we are regularly getting updated, and also we are working on those comments given by the working group for the development of this harmonization of the methods. So keeping all those in mind, we have come up with these methods where we have tested on different kinds of materials that belongs to multiple categories. So since the original recalls were on oil seeds, so you can see in this list you have sesame, which is the sesame seed, so sesame oil. Then you have, in terms of spices, we have curcuma, which is nothing but the turmeric that we use. Then we have methods on garlic powder, ginger powder, and other spices also. We are also working with FSSAI to come up with a harmonized method so that that can be used. It would be like a ready-to-use recipe, so the labs can easily adopt it. So here are some screenshots of how the results look like. So we are not going more deep into the technical aspects of how the methods are being set, but I'll just give you a general overview of how these methods are performing. So next, in terms of the application solutions that we have extensively worked on, is the PFAS, or perfluorinated or polyfluorinated alkyl substances. So in PFAS, these are compounds which have been there in nature for quite some time. And those who are organic chemists in the room, they know that the carbon and the fluorine bond is a very strong bond, and it's very difficult to break down that bond. So what happens is that when these compounds enter the environment, they stay there for a while. And when I say for a while, I mean for quite a while. So they have been there in the environment. PFASs were introduced very long back as coatings in non-stick ware. It is used in firefighting equipment. It is used as a water-resistant cover in textiles, and also in some types of protective clothing and jackets. It is also a part of, it's a raw material for many manufacturing processes. It's also the coating which is used on different kinds of consumer products. So you already have a lot of PFAS in your environment. So the PFAS development synthesis was done in 1930s, and it has continuously been exposed to the environment over the years, and now it is the 2020s that we see PFAS as a contamination or a problem in most of the materials that we come across. So the European Union specifically launched a program to monitor PFAS in different kinds of food materials, and what they found is that most of the fresh fruit, vegetables, a major chunk of it, as in more than 90% of it, passed. They were not showing PFAS residues. But when it comes to seafood, like shrimps, prawns, some of the fishes, there the PFAS rate of positivity of the presence of PFAS was quite high. So consequently, they have introduced regulations to control the levels of PFAS. So now the moment the regulations are coming into play, it means that we have testings also coming into play. So the European Union had released not only the regulations, but also some guidance documents, and these guidance documents have already been updated for quite some time. And there is also an annexure to this guidance document. So for those who are interested in delving deeper into what those guidance documents are. So it is more of the analytical requirement, and also how this individual compounds behave, what are the instrument methods, what are the minimum criteria that needs to be fulfilled. So this guidance document contains the details of those, and both the documents are more than 80 pages, so it's quite a read. And if we look at the number of publications and the way the testing market is also changing, you can see a renewed interest in PFAS. And here's a segmentation of the PFAS testing market. So if you look at the pie chart, you will see that most of the testing is done on drinking water and wastewater. So the major chunk is around 49% is in drinking water. And if you look at food, it is around 12%. But this percentage is expected to increase, and it's expected that in the next 3 or 4 years, this 12% will go and increase to around 18%, with the drinking water chunk still remaining the same. There are also some amount of environmental monitorings as well. And if you look into material testing, the REACH regulations also have some of the PFASs which are being tested on materials. But that category is, in terms of percentage or the chunk which is being tested, that is on the lower side. So therefore, now since the testing is increasing, there are some key regulatory drivers also. These names are very much familiar to you, like US EPA, FDA, so the European Union Chemical Agency, which formulates the REACH regulation, and also the Chinese Ministry of Ecology and Environment. They are taking into PFAS as a primary concern. And because of that, you can expect more number of compounds entering into the testing stream. So understanding the upcoming requirement and to understanding the upcoming challenges which might arise with PFAS testing, Agilent has invested strongly into this area, and we have come up with a complete workflow solutions. So this complete workflow solution includes the standards, which is the primary starting material. Then we have the sample preparation kits dedicatedly being manufactured for PFAS, because if you go back to 2 slides, then you would see that PFAS is almost everywhere. So most of the PTF labware that we use today is PTFE line, PTFE tubing, PTFE filters, PTFE containers, closure systems. Most of them have either a PTFE lining or a PTFE coating, or it itself is made out of PTFE. And PTFE is a major contributor for PFAS. So now we have a dedicated lineup of consumables which will take care of PFAS not coming in from your sample preparation. Then even on the instrument, we have PFC-free instrument kits. So the standard HPLC is modified to a PFC instrument kit. And then we also have a delay column, which is a hardware accessory that has been added to the instrument, so that the PFASs which are already present in the solvents that we are using, so that can be combated. Then we have dedicated vials, caps, of course, columns, the heart of any chromatography systems, and then the required software and the data analysis programs. So that makes a complete package for PFAS solution. So tomorrow when you are in requirement of a PFAS analysis, then when you have to comply to a regulation for PFAS, all you need to do is call Agilent, and we will help you set up the workflow. So these are for a glimpse, these are the PFAS PFC-free kits that we have. So there are some portions in the hardwares where the tubings, the bottle caps, closure systems, everything has been changed. And the portions that you see marked, highlighted in yellow, red, and blue are the ones which are changed in the hardware system. So the result is that we get an extremely nice chromatogram where over 100 PFAS compounds can be quantified and identified in one single run, which spans within roughly 12 minutes with an Agilent 1290 HPLC along with 6470 triple quad system. So, and this is the system that we have. We have extensive literature also already developed to meet the demands which are there for testing the shrimp, particularly seafood, and eggs or high-fat matrices. So we have already developed 2 dedicated workflows. One is for fruits and vegetables, which is low-fat matrices, and one is on shrimps and eggs, which is a high-fat matrix. And the range with which we can identify this molecule is from 0.1 ppb to 5 parts per billion. And you can see the big ships even at the 0.1 ppb. So for those not conversant with ppb, ppm, so to just give you a context, 1 ppb is equivalent to just 1 grain of sugar in a truckload of sugar. So the instrument is sensitive enough to spot that 1 grain of sugar in one full truck. So we have tested this across 18 different PFAS, the ones which are currently being talked about, the ones which are being stressed about by the different regulations. The methods are ready and ready to be transferred. And it also meets the current regulations which are there, and the instrument also has the potential or the capability to check these compounds or test for these compounds even at lower levels. So in the event of tomorrow, the regulation changing and becoming more stringent ones or accommodating more and more compounds, we are already there with the solutions. It's just a matter of time that we need to implement it. We have dedicated resources for PFAS. Similar to ethylene oxide, we also have dedicated application notes where we have focused specifically on certain EPA methods which are there for drinking water. We have also done the work for different seafood matrices, and there are also dedicated webinars which are there on Agilent webpage which you can check to understand basic of the regulations as well as the solutions that we have developed. Then another emerging application, not really an emerging one, this has been there for quite a while, it's that of the polar pesticides. So the challenges of ethylene oxide, the challenges of PFAS, and the challenges of these polar pesticides are all unique in nature. Why we have chosen to talk about these applications is that they represent different sectors, segments of the testing, and each of these segments are unique in terms of the challenges that they pose. So polar pesticides again are smaller molecules with highly polar nature. They are sticky compounds, they require a separate method, so they cannot be done by the conventional methods that we use. You may use a derivatization based method or a direct injection based method. There are multiple matrices in which we have to monitor these compounds, and of course robust separation and detection techniques to meet the low MRLs. So for this also the workflows have been developed. To show you an example, here we have 11 polar pesticides along with their internal standards which have been tested in tomato, which is a fruit matrix. And then we have the calibration curves across multiple concentration levels starting from 1 to 100 ppb. So it covers a huge range which is below the MRL as well as it covers a certain multiple above the MRL. So you can test for a huge range of concentrations which are present. So in short, for polar pesticides, we have methods for 11 polar pesticides which have been developed across 4 food matrices, across 4 different commodities, and these have all been verified and validated across with respect to the SANTE guidelines, which is again the rule book that is required for any type of analytical method validation that we use across in multiple testing labs. And this has also been evaluated on multiple systems. And the findings of this are published as conference posters. You can browse through the AOAC webpage to see these posters, or even on the Agilent webpage as well. So next online we are going to talk about something which has been there for quite a while. We are feeling the pangs of it, but not really making a shift, and that is helium. So for any lab which runs a gas chromatography system, helium carrier gas is of paramount importance. So we need a helium carrier gas to do the separation of the molecule. So whether it is a 200 molecule separation, or it's a 150 molecule separation, or even one single molecule that needs to be run on a GC system, we require helium carrier gas. But if we go back to the basic chromatography books, we see that hydrogen, nitrogen, helium, these 3 different gases can be used. Now, why we are talking about alternative carrier gases is that helium has a natural reserve and it's a finite reserve, just like we have a finite reserve for our petroleum reserves. So there is already a huge shortage of helium which is there, and you can see that those who are already into the lab procurement or have an idea about the procurement, they would see a delay in procurement of the cylinders. They would also see a rise in the prices. So as far as my experience goes, 8 years back we used to get helium cylinders within 15 to 16K per cylinder, and now the price is touching more than 35 to 40K. So the prices have gone extremely high with delayed delivery times as well. So there is a renewed interest again in getting back to hydrogen as a carrier gas. So because now hydrogen again has certain apprehensions. So hydrogen, as you all know, is a reactive gas. So there is an apprehension in terms of its safety as well. So for that, instead of cylinders, we can also use hydrogen generators. So supply is not an issue, safety is not an issue, it can be dealt with, it can be taken care of. But what needs to be more taken care of is the reactivity of hydrogen. Hydrogenation is a very common reaction which we all have studied in chemistry, the conversion of vegetable oil to vanaspati or dalda. So, that same reaction also takes place in our instruments as well. So when that happens and we are looking for analyzing compound A and we use hydrogen as a carrier gas, there is a high chance that we don't analyze compound A. We are testing for compound A while we are getting a report for compound B. So for that, Agilent came up with an innovative solution, and here we have developed a completely new hardware, which is a hydro-inert source that makes hydrogen inert to these compounds. So basically, hydrogen reacts with these compounds in the presence of a metal catalyst. And in all the mass spectrometry instruments, the ion sources are made out of metals. So that is something that we cannot avoid. The ion source has to be made out of metal. But what we have done is, we have made a new ion source which reduces the catalyst effect of this metal. And with that ion source, what we have done is, we have studied the efficacy of hydrogen carrier gas. Agilent was also the first vendor in the market to bring in methods with retention time locking, where we provided a complete database of 1,000 pesticides with their retention times for the users to easily implement those methods. Now, if we use hydrogen as a carrier gas, then what happens to the retention times? Everything changes. So it's like a nightmare for all of our users that we need to rework on the methods, we need to revalidate, and it's going to take another 3-4 months of our time with the instruments. So what we have done is, we have kept the retention times same. All we have done is, we have played with the dimensions of the GC column and brought the columns to in such a dimension that your database still remains valid. The MRM transitions also still remains valid. So the methods that you were using with helium are the ones that you will still be using, only the column changes and the carrier gas changes. So if you are using a 70-10 system or a 7000E system, all you need to do is switch over from helium to hydrogen, remove the 15 by 15 meter column, and then connect a 20 by 20 meter column. And again, we have validated this method completely on a variety of matrices, which involve difficult matrices like spinach, it involves matrices like spices. So, and we have seen that it also gives excellent sensitivity, which is enough to meet the regulatory requirements. And then again, why do we need this hydro-inert source? Is because if we test hydrogen as a carrier gas directly on a conventional ion source, we see a lot of change in the mass spectrum. So for those involved in identification of compounds, then it becomes difficult to search against the NIST library. So the hydro-inert source just takes care of that, and it reduces this conversion and thereby gives you higher matching scores. And another good news for our users who are there on the 7010 system is that the design of the 7010 system with respect to its lens sizes and openings has been made in such a way that the reactivity of the hydrogen with the compound in question is way reduced in the 7010 ion source. So you still get the same kind of NIST library searching. In fact, we even collaborated with one of our users from the fragrance industry, where they do a lot of testing on the flavors as well as fragrances, and they depend a lot on the NIST based searching to identify the compounds. So they did search for some of their products, and then we did see a very good agreement of the results with both helium and the hydrogen carrier gas. And there is also a detailed publication which is coming in from our Europe counterparts. So they have given in details about the different kinds of flavor compounds like that you have used as flavors in food manufacturing. So even there you can check the spectral fidelity of these molecules. So in conclusion, if you are planning to switch from helium to hydrogen, we are there to work with you. We have already worked on some of them and have established those methods. And all we are right now doing is that customizing it more towards the Indian testing context. So making it more specific or tailor-made to the requirements that we have in terms of the testing requirements in India. The same injection parameters can be used and the same sample preparation can be used. Nothing needs to be changed. So with that, what we have achieved in terms of the testing is that both for ethylene oxide and PFAS, we can meet the detection limits as well as for the polar pesticides. And we are also in the first stepping of turning the hydrogen carrier gas into a complete success story. So this can be successfully done with the help from most of our users, because they are also collaborating. Some of our users are also collaborating with us in this project, both globally as well as in India. So to see more about these applications and other applications that might interest you, so if you are from the beverage industry, if you are from the food manufacturing or the food packaging industries, then you would definitely be interested in food contact materials as well, like the extractables and leachables coming in from the food contact materials. So we have a dedicated page on Agilent, which is the Food Scientist's Vault, and it has free materials to access and download. Anybody can log in and download, and it is divided into multiple segments. So you can browse into your segment of interest, and from that segment, whether you belong to the food safety or food quality or food authenticity sector, so it could be simple testing for meeting the compliance requirement, or it could be an elaborate testing like metabolomics sort of study or finding out the markers. So you can go under these segments and look into the webinars and the videos which are there. So you can also follow us on our social media, where we keep posting about the different solutions that we come up. Our participants in the conferences that we have at the international platforms as well as in India platforms, and the events that we keep doing on. We also keep doing on a lot of webinars, education series for most of our users, and also on-demand series as well. So with that, I would like to end the talk for today. And if there are any questions, we can take those. Thank you so much for your attention.